What's in the Water by Bailey Schutza for the purpose of Chemistry 120N, Lab Section 2. Water is around us everywhere, and as all of us know, it is essential to human life. Therefore, the quality of water that we are consuming is incredibly important. The purpose of this lab was to identify unknown compounds in various drinking water samples. The sample I will be discussing is sample C, which is also known as vial 31. The two types of technology used were NMR and IR, spectroscopy. NMR shows the number of peaks and the ratio of areas, whereas IR shows the possible functional groups. Here is an NMR spectrometer as well as an IR spectrometer. Just to get more technical, the terminology for the technology used, so for the NMR, an Enzali instrument inked EFT60 NMR spectrometer with 1.4 Tesla magnet operating at 60 megahertz proton frequency was used. It had PNMR spectrometer control and data acquisition software that had one proton scan. And then lastly, NUTS Pro software was utilized to display the spectra. For the IR, a Thermo Scientific Nicolette IS5 FT IR spectrometer with ID7 ATR was used. It had a volatile sample cap, and it also had a diamond crystal ATR sample accessory that had a middle cap. And then Omnic instrument operating software was used, and it displayed the spectra, which produced 16 sample scans. For the procedure, first the IR spectra was taken. Functional groups that were present were recorded, and then the NMR was taken. The peaks and ratio of areas were displayed, so figures and tables were created for each of these, which were later used to compare to narrow down possibilities. As just said, they were compared, and then the unknown sample was able to be identified. So first, the IR spectra of all possible functional groups was recorded. So, for example, if you had an OH group and a CH group, you might have methanol, or you could also have ethanol. It just depends on your comparison between the IR and the NMR. So I used that previous table to compare to my graph that was produced for sample C. So as you can see here, this is an OH group because it is a broad peak in the 3500 to 3100 range. And then here we have a peak or a graph in the 3010 to 2800 range. So this is an OH group right here, and this is a CH single bonding carbon. So again, these results were compared to the previous table, and possibilities were narrowed down, as will be discussed in a little bit. Then the NMR was taken, and here we have a list of all possible number of peaks to ratio of areas. So this was used in comparison to my graph that was produced by the NMR spectrometer. So as can see, we have a 1:1 to 6 ratio, or 6 to 1 to 1, and there's 3 peaks. So this was compared to the previous table. So again, the IRs were compared, table 1 and figure 1, for OH and CH. The possibilities that I found were methanol, ethanol, 2-butanol, and 2-propanol. This was too broad of possibilities, so I needed to compare to my NMR in order to narrow it down. So the 3 peaks with a 6 to 1 to 1 ratio, the only functional group that contained that strict 3 peaks with 6 to 1 to 1 was 2-propanol, which was also a choice in my IR. So therefore, I knew that the resulting compound was 2-propanol. So some health risks from 2-propanol. So according to health risks when using isopropyl alcohol, we have when consuming and drinking water with 2-propanol in it, you might experience slowing of heartbeat, nausea, stomach pain, confusion, dizziness, low blood pressure, respiratory irritation, central nervous system depression, dilation of the blood vessels, vomiting, and disturbed motor responses. So as can see, it is pretty detrimental to health. And then some environmental effects, according to safety data sheet, it is harmful to the environment because of its high flammability and its mobility due to volatility. So what that means is that it's able to spread quickly throughout the water and it's harder to contain. When it's harder to contain, again, it spreads and it can get more into our surface area water as well as our drinking water. And then when using 2-propanol, it is advised to use electrical proof or explosion proof electrical, ventilating, and lighting equipment due to its hazards. And then just in conclusion, the quality of our drinking water is important when considering human health. So that is why IR and NMR spectroscopy are able to help us dictate what is safe and what is not. And then again, 2-propanol is just hazardous in drinking waters as well as surface area water. Here are my references, and thank you for your time.